Hello, I'm Adam, an engineer at Bio, the world's first drug-eluting branch stent to treat brain aneurysms. During this video, our team will explain how our product works, the key risks of our product, how we will conduct V&V testing to ensure safety, and how we will regulate our product. An aneurysm is a localized blood-filled balloon-like bulge in the wall of a blood vessel. A wide-necked aneurysm is a notoriously difficult aneurysm type to treat, defined as having a neck in excess of 4 mm or having a dome-to-neck ratio of less than 2:1. An aneurysm itself is not typically life-threatening. However, if an aneurysm ruptures, then hemorrhage and/or stroke can occur. Wide-necked aneurysms are currently predominantly treated with either an endovascular coiling surgery or a flow diversion stent. During endovascular coiling, a platinum wire is inserted into the aneurysm to occlude the area, prevent blood flow, and avoid potential rupture. Flow diversion devices have recently emerged as a method of diverting blood flow away from aneurysms to prevent aneurysm expansion and rupture. However, both these current solutions are not ideal and commonly result in side effects including localized inflammation and restenosis. To avoid these problems, we have developed Bio.
The Bio stent has two main features: the bifurcated flow diversion device and the eluted drug sirolimus. In our flow diverting stent design, we have incorporated an open region in the stent that allows for a second stent in the side branch. This design uses two catheters which are fed through the femoral artery, using balloons to expand the main stent. The second catheter feeds the secondary stent through the opening to be expanded by the balloon. The key advantage of the stent is that the stent is only placed in the parent vessel and avoids using a coil that can perforate the fragile walls of the aneurysm. The chosen material for the stent is nitinol due to its unique properties of superelasticity, shape memory, and biocompatibility. Secondly, our device is novel in that the drug sirolimus is used to reduce neo-intimal hyperplasia, that is, excessive growth of cells over the stent due to inflammation caused by the flow diverter. This lowers the risk of restenosis or obstruction of the vessel and further surgical intervention.
Bioproducts as a result of stent material degradation is one of the major contributors to stent failure and vascular inflammation. Nitinol was chosen with this risk in mind as it is biocompatible, and cellular rejection of degradation byproducts have not been found. Stent migration is another risk that will impede the functionality of our stent. We have incorporated a latch system to the ends of each stent component. In addition, the structure of our branch stents at the vascular junction will also help to hold each stent component in their respective positions. Neoatherosclerosis is chronic inflammation resulting from the rejection of the implanted stent and leads to subsequent neovascularization. This results in neoarthromatous neointima, which has been linked to the development of in-stent restenosis and acute thrombotic occlusion. Sirolimus has been shown to be a suitable eluting drug to reduce this pathophysiology compared to other drugs used in drug-eluting stents.
It is essential to ensure the safety and efficacy of our device with verification and validation testing. Initially, we will verify our device's mechanical and material properties with benchtop testing by following relevant standards. These experiments could include pulsatile fatigue testing, corrosion testing, and wear product analysis. The key relevant standards are shown on the screen.
So, how will Bio be classified? According to Rule 3.4 of the Australian Regulatory Guidelines, the proposed solution is surgically invasive, long-term, and is partially absorbed into the body. Therefore, it is a Class 3 medical device. Further, our drug-eluting stent combines a drug and a device, and is therefore regulated as a combination product under Section 503G of the Federal Food, Drug and Cosmetics Act. To gain regulatory approval, we will initially obtain a European CE mark by conformity assessment, then applying for an approval in Australia. The FDA is often a slower process. The clinical investigations of our drug-eluting stent will be conducted under an investigational device exemption. Then, a marketing application for the product will be reviewed under a pre-market approval application in accordance with 21 CFR Part 814. With adequate verification and validation of our design solution, we are confident that a safe and effective drug-eluting branch stent will revolutionize wide-neck brain aneurysm treatment. From all of us at Bio, we thank you.